Hello, everyone. Thank you for tuning in to this virtual presentation. And today I'll be talking about the tools we have at RBC that can be used for discovery of KRas pathway inhibitors. So before I start in my main topic, I'd like to take a moment to introduce our company and just give a very brief overview of all of our capabilities, since obviously KRas assays just a small portion of what we have to offer. So Reaction Biology is a contract research organization, and then we provide services in the areas of early drug discovery. So our company is almost 20 years old, and we have been steadily expanding our services portfolio over the years, starting with kinase profiling assays from 2006, and then we added epigenetic profiling services in 2010, and then we added ion channel screening platform in 2018 through acquisition from Jubilant, and also last year we've acquired ProKinase, a company that has a lot of experience with kinases and many interesting in vivo capabilities. So currently, reaction biology can offer various services such as biochemical, cell biology, and biophysical assays, in vivo testing services, as well as custom protein production and ion channel assays. And obviously, I don't have time to go through all of these in detail. I do invite you to visit our website to get more information, or email us directly. We'd be happy to provide you with more info. So if you're watching this presentation, you probably don't need a detailed introduction about KRas signaling pathway in disease, but I'll give a little bit of background in order to put things in perspective. So RAS signaling pathway transduces extracellular signals into intracellular responses, as shown here in a very simplified diagram. And then KRas belongs to a family of small GTPases, and there are a couple of homologs, HRAS and NRAS as well. And then all of these are known oncogenes that are frequently mutated in a large percentage of cancers. And then problems in RAS signaling contribute to about 20 to 30% of all human cancers, and about 90% of pancreatic cancers, for example, have mutated KRas. And then all of it is associated typically with poor disease prognosis, and then in disease state, aberrant activation leads to increased signaling through the pathway that promotes cancer development through a variety of mechanisms. So KRas itself exists in two forms, inactive and active. So when it's bound to GDP, the protein is inactive. And then as GTP binds to KRas, the conformational changes occur that enable interaction with effector proteins that in turn lead to signal pathway activation. So this cycling requires additional proteins. And then in order to go from GDP to GTP, guanine nucleotide exchange factors are necessary. And then even though RAS itself is a GTPase, and intrinsic activity is very low, and then GTPase activating proteins, GAPs, are required in order to increase the rate of hydrolysis. And then mutants, particularly glycine 12 mutants of KRas, are typically locked in this GTP bound state due to impaired GTP catalysis. And all this leads to increased signaling through the pathway, and then the negative effects that are associated with it. So KRas has always been a desirable drug target, and then for a long time it's been considered as undruggable due to the absence of a well-defined binding pocket. And then there's also very high affinity to GTP, so then making GTP competitive molecules is pretty much impossible. And recently there's been a resurgence of interest in KRas inhibitor discovery, partially due to the fact that there are several new binding sites that have been identified, such as a shallow pocket between switch 2 and switch 1 regions, right about over there. And then also there were several compounds identified that actually bind covalently to G12C mutant, and that happens in a switch 2 pocket. And in addition to targeting KRas directly, different alternative approaches are also being currently explored, such as inhibition of exchange factors, inhibition of interaction with effector proteins, as well as inhibition of kinase activity, of course, and as well as some others. So over the past couple of years, we've produced a number of proteins, and then also validated a set of assays for the discovery and characterization of compounds that target different steps of KRas pathway. That's what I'll go over briefly today. So we began with production of recombinant KRas proteins, and then we've made several constructs, mainly with different affinity tags to enable downstream assay development. And then the sequence is the same in all of them. They do exclude the hypervariable regions since it's required for membrane localization and not really necessary for in vitro assays. And then currently we have wild type and then 3 G12 mutations, and then also working to produce additional mutants in the future. And of course, we need factors and effectors in order to set up the assay. So we did make both SOS1 and SOS2 protein, and we produced one GAP protein, RAS1, as well as RAS binding domain of CRAF protein. And as you can see in the graphic, the target proteins are quite large. So we just focused on the relevant domains that are needed to get certain activities. And then the amino acid boundaries of the proteins that we currently do have, they're noted under each protein graphic. And then also all of these proteins are available as protein products. So if you would like to get them and set assays up in your own lab, they're available for purchase. So hard work of many groups produced a number of tool compounds that are available, and then they target various steps of KRas cycle. And then for our assay validation, we mainly used 4 molecules shown on the slide. So 2 compounds on the left are specific for G12C mutant, and they bind to the GDP version of RAS, and then they lock it in this off state. Then BA293 compound is specific for SOS1, so it prevents nucleotide exchange through this protein. And then finally, BI2852 compound, it binds to RAS, and it prevents interaction with both SOS1 and then CRAF protein. And this compound is not specific for either GDP or GTP bound state of KRas, and also it doesn't matter what mutation status of the protein is, it'll interact with all of them. So I will start with nucleotide exchange assay, as it's pretty commonly used to identify compounds that will lock RAS in the GDP bound state. And it monitors SOS-mediated exchange of fluorescently labeled GDP to GTP. And then this reaction can actually be performed in the absence of SOS, but in this case, you need to use quite substantial amount of EDTA, since the unmediated exchange reaction is pretty slow, as shown here in red. It's pretty much linear, nothing changes. And then there's several options available for use for GDP, fluorescently labeled GDP, so either MAD GDP or body GDP. And then the whole assay is based on the fact that when fluorescent GDP is bound to RAS, its fluorescent level is higher, and as it's being displaced, the fluorescence decreases, as you can see here in green. And then in order to get the rate of this exchange, we can use exponential decay equation and get it that way. And then this figure here on the bottom just illustrates the activity of our 1620 compound. For example, at 1 micromolar, the rate of exchange is pretty much equivalent to no SOS. All of the protein is actually locked in its GDP state and cannot exchange to GT. So as I mentioned, main application is obviously for screening compounds in order to deactivate the exchange to GTP. So on this figure on the left, we're looking at 2 compounds with wild type and G12C mutant. And as you can see, they have no effect on nucleotide exchange of the wild type, and then they nicely inhibit G12C as expected. And then the figure on the right illustrates the utility of the assay as a selectivity profiling tool. And so here we're testing wild type and then 3 mutants with KRPEP2D compounds. It's a peptide-like inhibitor for KRas. And as expected, they do inhibit nucleotide exchange in all 4 variants, and then it's most potent against the D mutant, as expected. So we've also worked out several assay modifications that enable additional application of this assay. So this slide illustrates a modification for the assessment of time dependence. So what we've done is increase SOS1 concentration so that the rate of exchange is very rapid, as shown here on the left. And then so you can see that under the setup, but pretty much all of the GDP is exchanged to GTP in about 2 minutes. So by using these assay conditions, we've tested Amgen 510 compound with various pre-incubation times, and we can show that the IC50 decreases to about 10-fold. So this is something that you could expect for covalent compounds. Alternatively, if we decrease SOS1 concentration, we can observe a linear rate of nucleotide exchange, and in this case, such setup would be actually more optimal to look for SOS1 specific compounds, since the SOS concentration is quite low. And then again, we tested all 4 variants with BA293 compounds, and indeed we see the effect on all of them. And then some variation in IC50 is likely due to different SOS concentration that have to be used in assay with different mutants in order to get sufficient window. So because nucleotide exchange will both work with both SOS1 and SOS2, this could be used as a selectivity tool as well. So here we're testing BA293 with SOS1 and SOS2, and obviously it's only working with SOS1 and has no effect on SOS2-mediated nucleotide exchange. And in comparison, BI2852 compound, since it binds to KRas, it will inhibit both SOS1 and SOS2-mediated exchange as shown with also wild type and the mutant. An alternative to the assay I just talked about is HTRF-based nucleotide exchange assay. So in this case, the concept is pretty similar, but we're using time-resolved fluorescence to detect binding of GTP, which is in this particular case is fluorescently labeled in comparison to GDP in the previous example. And then we expect signal to increase as labeled GTP binds to KRas. So this is a good alternative in cases when there is fluorescent interference of compounds that we sometimes observe, especially with MANT-labeled GDP. And then another difference is that this assay has to be performed at lower submicromolar GTP concentration just because the molecule is fluorescent, so it needs to be minimized. And then the assay works comparably to the other one, and it can evaluate various modes of nucleotide exchange inhibition. So here is illustrated with the C and V mutant, and then all 4 compounds work nicely with the C mutant, and then only 2 compounds work with the D mutant. Now I'm going to move on to a different assay type, specifically disruption of protein-protein interactions. And in the first example, we're looking at the interaction of KRas and SOS. And this is a good orthogonal method to a nucleotide exchange assay if you're looking for SOS1 specific compounds. So detection is again HTRF based, and then in order to see interaction between 2 proteins, RAS has to be GDP bound. So then the compound is expected to prevent this interaction, and this is indeed what we observe using either mutant or the wild type protein, and the effect of compound is pretty similar with both. So a similar concept is used in this case, so where we're doing disruption of protein-protein interaction, this time with effector protein, CREF. And then in order to see some interaction, the KRas has to be loaded with GTP. So in this figure, you can see that this binding is more robust when the protein is GTP loaded. And then by testing BI2852 compound with either D or C mutant, we see very similar effects, so interaction is disrupted. And then here on the bottom, we can show that RS1620 and Amgen compound have no effect on this interaction as expected. Most likely, they just cannot interact with KRas when it's in the GTP bound form. And then this CREF binding has an interesting other application. We can actually use it to detect nucleotide exchange. And then it can be viewed as sort of this all-in-one assay, since the effect of all steps of KRas cycle can be seen in this one single reaction. And then we can also use it to look at SOS1 independent GTP binding without using EDTA this time, even though this reaction is much less efficient when SOS is present. There's still enough assays sensitive enough, and we get enough signal in order to evaluate compounds. So overall, different versions of nucleotide exchange assay that we've tried generate very similar IC50s. So there's not really a best assay that I can recommend. It's pretty much what you like best, and anyone can be used to study compounds. So now moving on to biophysical methods that we have available for KRas and SOS. So we've set up a thermal shift assay, and this assay is based on the fact that if compound measuring the melting temperature of the protein. So if compound binds to the protein and stabilizes it, you would expect to see an increase in melting temperature. So this is exactly what we observe as shown here. Covalent compounds bind to G12C mutant and stabilize them, and then the TM changes quite significantly, and they have no effect on the other 3 variants. And then, surprisingly, we didn't really observe very significant shift in TM for BI2852 compound in this case. And for BA293, it does shift TM of SOS1 and then has no effect on SOS2 as expected. So thermal shift assay is sort of more good for initial screening, since you cannot get any binding constant for the compounds. And then if you're actually looking for very detailed kinetic analysis. Obviously, SPR is the technique of choice, and we do have it set up for both KRas and SAS. So when performing SPR assays, typically the protein is immobilized on the surface of the chip, and then the compound flows over. And then as the interaction occurs, the response is measured, and then based on the response, you can measure association constant, dissociation constant, and then using these, the compound affinity can be calculated. So just an example of some data for KRas, and for this assay, we're using biotinylated KRas. So we measured binding of KRPEP2D, so peptide compound, to all 4 variants, and as expected, we see interaction with all of them, and the highest affinity is observed with G12D mutant. So as I mentioned, we also have SPR assay available for SAS1 protein. So this slide shows the binding sensogram and then also fit for BA293 compound to SAS1. And then we observe a KD of about 7 nanomolar in this case. So we have 2 Biocore 8K instruments available in the lab, and then we're actually looking to purchase 1 more in the near future. And these instruments are great for library screening due to their throughput, and then obviously you can also do binding kinetics. And then in general, SPR is a good method to look at binding of small molecules, but not only, also you can look at peptides and then protein-protein interactions. So of course, I cannot finish the presentation without mentioning kinases and inhibition in kinases in particular, since these are actually the proteins that get activated downstream of KRas. So RAF and PI3K are the 2 pathways, are 2 best characterized effector pathways, but there's obviously many more kinases that are associated with just big KRas signaling pathways. And then inhibition of a single kinase in vivo could lead to activation of compensatory pathways and then the development of resistance phenotype. So it is beneficial to have compounds that you're able to inhibit more than 1 kinase at a time in order to prevent this. So reaction biology has the largest collection of kinase assays. We actually have over 700 proteins available for selectivity profiling and screening. And we use radioisotope assay, so P33 ATP as a substrate. So that allows us to look at direct phosphorylation of substrate without need for any modified substrate or coupling reagents. So detection is obviously very sensitive. And then there's also very little interference of compounds with the detection, which is sometimes the case with fluorescent detection methods. And then before I finish my presentation, I want to mention that we have a growing collection of kinases that have been validated in kinase cellular engagement assay. So we're using the nanobread assay from Promega, and it's based on bioluminescence energy transfer. So what you can use this assay for is to look at parent binding affinity of compounds in the cellular context, as well as cell permeability to test compounds. It is also HTS compatible, so large libraries can also be screened using this method. So kinase itself is fused to a luminescent reporter, nanoluc, and then each kinase has a fluorescently labeled probe. And then both probe and compound are added in the cell media. And then as they penetrate the cell and compete for the same site on the kinase, you observe a reduction in bread signal, as illustrated in this one example for AKT1 kinase and its control compound. So we currently have 170 targets already available, and then hoping to reach 200 by the end of the year, and then keep growing from there as they become available. So at this point, I would like to finish my presentation, and then thank you for your attention, and I would be happy to address any questions that you may have. Thank you very much.